Hello and welcome to the Beyond Biotech podcast number 54. I'm Jim Cornell from Labiotech, and I've been busy trying to think of something that happened this week that wasn't either mundane or weather related. And as I really didn't do anything because of the rain, that means I'm reduced to details about today, which is July the 14th. It's a holiday in France, and it's also, for some reason, National Tape Measure Day. Those things that you need twice a year and have absolutely no idea where you left them. I'm sure there are people who use tape measures much more frequently, but for me it's not that often. And also I guess I'm not very organised, otherwise I would just put them in the same place and remember where they are. It's also Shark Awareness Day today. I assume that that's the ocean creatures and not the people that try and scam you. Anyway, this week we have an interesting subject on the podcast. We are chatting with Matthias Scheier, CEO of TolerogenixX, about some great results for its cell therapy treatment of transplant patients. So more on that in a little while. That means it's now time for the news you may have missed over at Labiotech.eu. We had a special newsletter on mRNA technology that included 5 innovations to take RNA vaccines to the next level, mRNA for transformative medicine, ready-to-use enzymatically produced cell-free synthetic DNA for mRNA manufacturing, inhaled mRNA medicines, and quite a few more. We had an article on 5 advances in lung cancer research. An existing cancer drug could be repurposed to fight certain aggressive cancers, and NKIO was awarded a £1.6 million grant to develop natural killer cell therapy. NodThera is the first company to show a reduction in neuroinflammation with an inflammasome inhibitor. Immune memory research could lead to the next generation of epigenetic drugs, and there was an article on AIS Biotech, a startup using carbohydrates to fight the flu. Crossbow Therapeutics launched with $80 million to target cancer. Pfizer invested $25 million in CellCentric, and ADC Therapeutics paused a clinical trial following patient deaths. We had an article on Medicon Valley in Denmark and Sweden. A new venture capital company, Biovance Capital Partners, was launched, and French gene therapy company EG427 raised €18 million. A partnership has been formed to progress neurodegenerative disorder treatments. TME Pharma provided some positive glioblastoma trial results. Calliditas announced encouraging results from its head and neck cancer trial, and Biogeneration Ventures closed its BGV Fund 5 at €150 million. We had an article on financing, another on how AI has discovered an existing drug works to cure a rare disease, and you can read all of these and more at Labiotech.eu. And that means we're into today's interview on solving issues related to the rejection of organs after transplants. To tell us about those issues and what TolerogenixX is doing in the field using cell therapy to hopefully make this a thing of the past is Matthias Scheier, CEO of TolerogenixX. I guess the easy first question then is if you could give me a bit of background on the company.
Yeah, we found a company in 2016. It's a spin-off of Heidelberg University. We are 3 founders from an academic background. My name is Matthias Scheier. I'm the CEO of TolerogenixX, and I have worked in the field of tolerance induction since 20 years roundabout. And my co-founder, Christian Morat, I know Christian from our doctor thesis, started in 1998. We worked in the same lab, and then we collaborate since the doctor thesis now for many years together. And Christian is our CSO. Christian Morat is expert in the field of transplantation, and his scientific background is focused on this area. And Anita Schmidt, she is our CTO, our Chief Technology Officer. She has a lot of experience in the field of manufacturing of ATMP cell therapies and bringing such new cell therapies from a basic science lab to a GMP core facility with all the approvals and so on. And then we start first with a grant from the German government. This was our first money for developing this new approach. And then with this money, we made the technology transfer. We planned the phase 1 study, and then we start and do the phase 1 study. And at the end, we had a seed investor, the High-Tech Gründerfonds from Bonn. And after follow-up of the study, the TOL1 study, we worked in the field of deceased donor as another approach for our cell therapy. At the moment in the study, we work with living donor transplantation, but it's possible to transfer this technology to deceased donors too. And this is another possibility for our approach. And beside the transplant area, we have a lot of potential in the autoimmune area. In the autoimmune area, we have done so far preclinical studies for multiple sclerosis and systemic lupus erythematosus.
As far as the transplantation goes, could you tell me about some of the issues and the challenges with addressing that?
Challenges, our first challenge was to get approval for clinical studies, but we have already in place very easy manufacturing and very safe manufacturing process. And therefore, this high hurdle was not too high for us to manage it. The next larger step was to do the phase 1 trial, the first study in humans. Yeah, but this was not so hard to find patients because it's for donors and recipients. It's not a big issue to do this cell therapy. For example, for the donor, it's only an unstimulated leukapheresis with small needles in the arms. It needs around about 3 or 4 hours. And we collect the white blood cells. And then the white blood cells goes in the clean room. And on the same day, it's only a half a day procedure. The recipient gets the modified donor cells. And for the recipient, it's not a hard stuff too, because the recipient receives only the cells without any co-medication. And so far, we see no side effects in the recipients or in the donors. And therefore, it was not a big challenge to find patients which want to participate to the study. The harder problem was to find funding, because this indication is not so in the scope of big pharma and big venture capital firms. And therefore, we need a little bit time to find investors which are interested in this field of medicine.
Right. And the treatment that you give to people, is that just once, or do they need to have repeat treatments?
It's only one treatment in the field of transplantation. It's one single infusion with modified donor cells. In the field of autoimmune diseases, so far, it's difficult to test this or to examine this with animal studies, and therefore we have to wait since we have clinical data. In the autoimmune field, it could be that you need repeat infusions. For example, after 2 or 3 years, you have to repeat the infusion. But in the field of transplantation, we have now more than 5 years of follow-up data, and we see that all the parameters are in place. The patients have excellent graft functions, no signs of any rejections, and no donor-specific antibodies. Normally, you see this in our recently published 5-year follow-up data, that normally around about 20 or 30% of the recipients develop donor-specific antibodies, and then the end, these donor-specific antibodies end in a humoral rejection and in a graft loss at the end. And our patients treated with the Mixal, we have no signs of donor-specific antibodies. And this is very remarkable, I will say, because we treat the patients with 10 of the power of 10 Ferregen cells with Ferregen antigens, and we see no donor-specific antibodies or no antibody reaction. This shows us that this is a completely new mode of action, which works in a totally other way.
Before your treatment came along, what were the treatments before that?
Yeah, so far the common gold standard is a multi-track based immune suppressive therapy, which includes a calcineurin inhibitor like tacrolimus or cyclosporin and mycophenolic acid product, and steroids, and in the beginning an induction therapy with ATG or with a product like Similac and IL-10 inhibitor.
Are there other companies working on other ways to treat rejection?
Yes, there was in place there is in place one of our main competitor, Talaris. I think you know Talaris. It's a stock-listed company from US. Their approach is to combine the bone marrow and the kidney transplantation. That means the donor donates some kind of bone marrow cells, and then you transfer a new immune system. That means at the end you replace the immune system. But so far they have stopped their freedom trial because of side effects, and stopped their transplant program because of side effects. And because this for the recipient, that means at the beginning severe immune suppressive therapy, chemotherapy and radiation with a lot of risks at the end. And in the phase 2 study, there was some severe side effects, and now in the in the phase 3 study 2, and therefore they stopped their trial in spring this year. This was our main competitor, and was the competitor with the best funding so far. And then there are some other approaches to induce an personalized immune modulation, or to induce tolerance. For example, there is Quell Therapeutics with the CAR-T-REG approach, but they have, so far I know, not this clinical data. They have animal data and in vitro data so far, and they cannot treat all patients which wait for a kidney. They need a specific signature in HLA antigens. So far, I know. These are the two main competitors at the moment, I think so.
Obviously not giving any secrets away, you mentioned that you take white blood cells from donors. How does that work when it goes into the body of the recipient?
Yeah, how does it work? All cells within core have the HLA antigens on the surface. And the white blood cells are very easy to get, and they comes after we take them out with the leukapheresis, and then next few hours you see no changes in the blood results. And it's very easy to come to this HLA immunological fingerprint barcode with this tool, the white blood cells. And then we change on the blood cells with our technology, the co-stimulatory molecules from an aggressive phenotype to a tolerance-inducing phenotype. We change the flags on the white blood cells. That means at the end we can rise up white flags, and then we infuse this modified donor cells with the HLA barcode and the white flag to the recipient. And then this white blood cells goes into the spleen. The spleen recycle the white blood cells of the donor, and then structures of these mixed cells comes in contact with immature immune cells of the recipients. And then this immature dendritic cells, for example, develop to tolerogenic dendritic cells. And then in the end, we find very high levels of regulatory B cells. In our patients, we find 65-fold higher levels in regard to the B-regs in comparison to the normal control patients, transplanted patients. We can induce with our mixed cells specific regulatory B cells in a very, very high level.
Is this something that is easy and cost effective and can be scaled up?
Yes, absolutely. Because you have a massive cost reduction in the traditional multi-drug therapy at the end. You have a cost reduction of around about 80% of the standard treatment maintenance therapy. And this means our therapy is at the end cost effective, and it's cost saving for the payer. And this only in regard to reduction of infections, reduction of rejections. And now with the phase 2b study, I think we get better database because our last pharmacoeconomic results were based on the results of the phase 1b study. I think with the phase 2b study, we can show better the benefits of the MIG therapy in regard to quality of life too, in regard to hospitalization. So far, the patients with the classical multi-drug therapy 2 or 3 times per year in the hospital because of infections. And with our only so far 3 to 10 patients in the phase 1b trial, we could demonstrate that we have lower rates of opportunistic infections in contrast to the control group. That means now with reduction in maintenance immune suppressive therapy, you see that the patients have fewer infection rates in regard to CMV virus, polyoma virus, severe pneumonia, and so on. And I think with the now ongoing phase 2b study, we could demonstrate these benefits and could confirm these benefits of our MIG approach.
Do you have the opportunity, or do you get to hear from patients that have undergone this therapy? Because they must obviously be delighted to not have any of these issues.
Yeah, I'm as the sponsor, I have not direct contact to the individual patients, but I know from the study sites that the patients which are included in the studies are in a very good conditions, and so far we have no severe side effects reported. But I'm unfortunately not the direct contact to the patients. But I think if you have no steroids in your treatment, if you have no mycophenolic acid in your treatment, then you will feel a benefit, because this immune suppressive drugs, they have side effects, and therefore now in the B arm of our studies, the patient have only one drug in the maintenance therapy, in comparison to 3 drugs in the immune suppressive drugs in the maintenance therapy. And I think they make a difference for the patients.
And you said also that it's not just in transplantation patients that this has applications. What are the other applications that this could potentially be used for?
At the moment, we are only in the transplant area in humans, and now we make a pre-treatment in transplantation. That means 1 week before transplantation, the recipient received the mixed cell from the donor, but we see potential to treat already transplanted patients later on. For example, if they have severe rejections, we have done so far 2 individual treatments in patients with rejections, one with a cellular and one with a humoral rejection, and this works very good too. That means at the end, we see potential for patients already transplanted to treat them with modified cells from the donor or with similar white blood cells from a donor bank. And we see potential in the field of autoimmune diseases too, in the field of MS or SLE. But so far in this area, we have only animal data.
Where do you go from here, I guess? At what point does this become something that's just routine?
Yeah, I think we think that we have a good chance for conditional approval after the phase 2b study, because it's in an orphan disease, because it's an ATMP therapy, and then we could aim for an approval in the year 2026 or 2027 roundabout. And then it with good luck and so on, then it's available for patients.
And are you pursuing that in different geographies?
Yeah, so far the study runs in Germany, but the next step would be to bring the therapy to first to the Eurotransplant region, and then to whole of Europe, and then to US, and then to Asia, step by step. So far, our MIG therapy at the end is approved for 24 hours transportation. That means after manufacturing process, we have 24 hours to bring the cells back to the hospital where the patient is. But we know from our in vitro data that the stability is longer. It's around about 2 days, but with 24 hours with modern logistic, you need not so many hubs. That means for example, one manufacturing site for Europe is sufficient to bring the cells to whole of Europe. Another hub in US makes sense, and one hub in Asia. But if the studies comes to a good end, what we believe, then we will start, for example, next year to prepare all the things for market entry, and to do a technology transfer to manufacturing partners and so on, to have enough capacity to bring the cell to all the regions in the world where the patient needed.
Are there any other things in the pipeline of the company, or is it really this that you're just concentrating on?
At the moment, we are concentrated on the phase 2B study for living donor kidney transplantation. And then in parallel, we work for starting a trial in autoimmune diseases with a focus on SLE. We have done some animal models so far and some in vitro studies, and now we want to go to human blood samples and to make some final experiments with bloods from SLE patients and to have the whole package ready for scientific advice with the authorities and for preparing a protocol for the next clinical study, then for SLE patients.
You mentioned obviously this is working for kidney transplants. Would it have the same positive effect for other organ transplants or for things like graft versus host disease?
Yeah, we know from our animal studies in the animals, we have tested different models for transplantation, liver in pigs, heart in rats, hind limb in rats, and now skin transplantation in mice, and it works in other field of transplantation too. Kidney is very challenging in regard of immunology. That means at the end, if it works in kidney, then it works in the other solid organs too. Liver, for example, is the easiest one. And if it works in kidney, and kidney is the biggest market in the field of solid organ transplantation, and then liver, and if it works in kidney, then it will work in liver and so on too.
Hopefully good news there for the future of organ transplantation. So thanks a lot for joining us for this week's podcast, and I hope wherever in the world you are, you have a great week ahead. Take care, and you'll join us next week for another Beyond Biotech.